Good morning, everyone. Welcome to Viruses and Vaccines. I'm Helen Chu from University of Washington, and today we have two sessions. With the first having JT Macron introducing the providing the SARS-CoV genomics update, followed by a talk from Professor Addie Stern from Tel Aviv University. And just a quick reminder to go ahead and put your questions in the chat. And then I also just wanted to let people know about the speaker for next month, who is going to be Professor Akiko Iwasaki from Yale University and HHMI, and she'll be speaking about long COVID, and that's in January, on January 16th at 9 a.m. So I'll go ahead and turn it over to JT to start, and then stop sharing.
Thank you. Thank you, Helen. Yeah, so just a quick update, some review from last week or last month when we met, and then where we're headed now. So just to recap, when we talked last time at the early November, wastewater indicated that cases were on the downtrend. We had a bit of a wave over the summer and the fall period. We talked briefly that had been caused by this epistatic accumulation of mutations in the XBB sublineages. So we had this mutation at site 456 in spike, which then allowed this further mutation at site 455, and you had this progression from green to blue to orange. And we had seen this progression a number of times, and so this was indicative of positive selection. And indeed, if we look at the frequency of those mutations over time, first a mutation at site 456, followed by the double mutant, we saw those frequencies increasing throughout the end of the summer and into the fall. And we saw this sort of around the world and multiple times in multiple locations. At the end of our last presentation, Trevor mentioned that there was a new variant, JN.1, and we'll get into that a little bit. What we see now is that cases are on the rise, right? So wastewater showed that cases were decreasing, and now we're seeing them increasing quite steeply, nearly reaching peaks that we saw in the last winter period. And again, we hinted at that this is caused mostly by a new variant being driven. Here, this is shown now in yellow. So we see the single and the double mutation that we were talking about before on the EG.5.1 background, and other lineages are now decreasing in frequency, whereas this novel mutation at site 455, the same site as we saw the double, is now increasing quite drastically. And when we look on the tree, just to orient you, these are again the blue to orange progressions we saw before. The yellow mutation, this 455 mutation, falls on a divergent lineage. So it falls on the BA.2.86 background. This is a very long branch. This lineage was first detected over the summer and caused quite a stir because it had a huge number of mutations, both in spike and throughout the genome, similar to what we saw when Omicron first came out. This mutation, or this lineage, didn't spread too quickly at first. So this is showing some work from the Bedford lab here, growth rates of BA.2.6, growing fairly slowly, just sort of persisting in a couple of locations. However, once we add this mutation at site 455, we see rapid growth across multiple countries around the world. So this is now BA.2.6 plus this mutation is now called JN.1, and we're seeing consistent growth rates across multiple countries, which again is indicative, right, of positive selection, immune evasion, and a fairly robust signal. So this is the fastest growing lineage that we're aware of at the moment. There's been some work by the Cow group in Peking University to characterize the antigenic escape of this variant. So you can see here we're looking at neutralizing titers from individuals who were vaccinated 3 times, and they had a BA.5 or BF.7 breakthrough infection, and then an XBB infection. So you can see here, this is the parent lineage, this long branch lineage, BA.2.86. Its neutralization titers is up here, and then you see quite a drastic drop with this addition of a single mutation. This mutation came at a cost of ACE2 binding affinity. So that's shown here. BA.2.86 had quite good ACE2 binding, stronger than some of the other variants that we've been more familiar with over the fall and summer. But then again, with this addition of the mutation at site 455, we get a loss of ACE2 binding affinity. And so there's been this trade where the virus has sacrificed some binding affinity for further immune evasion. And actually, you can see that that while BA.2.86 is antigenically distinct from these other lineages because it's so far diverged, it was still susceptible to a particular class of antibodies, and that this single mutation allows it to avoid that class of antibodies, and is likely responsible for its immune evasion and rapid growth that we're seeing. So I think there's a big question of, okay, this is every time there's a new variant, how do the vaccines do against the variants? There's a recent preprint looking exactly at this question across a number of different individuals or classes of individuals who have had different immune histories. So one is just looking at the XBB.5 monovalent vaccination. Over here, we have people who have had XBB infections recently in the C panel, and then D is the individuals who had a recent Omicron infection followed by the monovalent vaccination. And so you can see in each case, JN here, that vaccination or exposure to XBB boosts neutralizing antibody titers against JN.1. But you can also see that these antibody titers are, again, lower than what we see for other circulating variants. And so, vaccination is better than not vaccination at the individual level. At the population level, we still expect JN.1 to continue to have this immune advantage and likely to continue to spread until the next variant arises. And I think that this sort of example of what we've seen over the fall into the summer now really highlights sort of the two paradigms of SARS-CoV-2 evolution, where on one hand we have this almost regular cadence of antigenic mutations that feels expected, very rapid, but expected, where you have accumulation of mutations within spike giving you some immune escape. And then every once in a while we have these very long branch events where a new variant seems to come out of nowhere. And although this happened right with BA.2.86 most recently, it happened to a certain extent with XBB, it certainly happened with Omicron. It still feels, at least to me, unexpected and wildly unpredictable at when that happens. And so I think what we have here is we have, immune evasion developing in the population level, and then we also have the impact of these long chronic infections likely driving global dynamics. And so that's part of the reason why I'm so excited for our speaker today. And then thank you. Yeah, so sorry. Just I can take any questions in the chat, and thank you. Of course, this data and all these results come from the GISAID and a lot of efforts by individuals around the world. So thank you very much.
Okay, I can. Yeah, thank you so much, JT. So it's my pleasure to introduce Dr. Addie Stern. Dr. Addie Stern is professor of virus evolution at Tel Aviv University. She received her master's and PhD in phylogenetic modeling, also from Tel Aviv. She completed 2 postdoctoral fellowships. At the Weizmann Institute, she studied microbial phage coevolution. At UCSF in Berkeley, she studied experimental evolution in viruses. In 2011, she was awarded the UNESCO L'Oreal Prize for Women in Science. She's contributed significantly to theoretical and experimental understanding of RNA virus evolution. And I'm particularly excited about reading the preprint that the talk is based on. It seemed like one of the first attempts, or the first kind of systematic attempts to really understand what's going on with these chronic infections in this very clever way. And we see how important this is for BA.2.86 and so forth, and really understanding what's happening here is hugely important to figuring what's going on with SARS-CoV-2 into the future. So I'm really looking forward to Addie's talk, and please go ahead.
All right, so I'll just share my screen quickly. Okay, so I hope everyone can see properly. Okay, so thanks a lot, Trevor, for that wonderful introduction, and thanks to Trevor and Helen and everyone for this opportunity to tell you about our work. I'm sorry, I'm just recovering from some cold, which is going to cause me to cough a little bit. Right, so the title of the talk really is using big data to identify chronic SARS coronavirus 2 infections, and how I hope this will allow us to sort of use this as a window into looking into future evolution. Before I begin my talk, I'd like to thank the people who did the work, the 2 fantastic PhD students, Sherry Harari, who's actually currently a postdoc at the Fred Hutch, working with Jessie Bloom, so she's right nearby to you guys, and Danielle Miller, who's going to be graduating soon. And this was a joint collaboration with David Burstein at Tel Aviv University, and with Shai Fleischon from the Israeli Ministry of Health, who was actually one of the first people to detect BA.2.86 and sequences from Israel. But before I really go ahead with the talk, I feel I can't sort of not talk about what's, just for one very brief second, about what's going on here in this really conflict-torn region where there's so much suffering going on as we speak. And this is my group that you see here. Everyone here has experienced some loss of some sort. And I'll just highlight two students actually are both continuing this project of SARS-CoV-2 chronic infections. So Yael on the right is a master's degree student. Four of her very good friends were murdered at the Nova Peace Festival, which took place on October 7th. And Nofar, the student on the right here, her brother was abducted to Gaza. So this is him, Yagev and Rimon, so her brother and her sister-in-law were abducted into Gaza. They were kidnapped from their homes. These are not settlements, at gunpoint. Rimon on the left was released on November 28th, and Yagev is still held hostage in Gaza for more than 70 days. And this is Nofar, my student, as you can imagine, is not doing any research whatsoever and just trying sort of almost helpless attempts to get her brother back home. So I really felt I couldn't not say anything about this while this is going on here. And in this time, in these 2 months where all these horrible events have been taking place, I found out that science is really therapeutic. And so let's move on to my therapy and talk about science and what we've been doing. So I don't think you really need an intro on SARS-CoV-2, but just to get us on the same page, we have right. This is an RNA virus. The genome length is about 30,000 bases. We have the spike gene, which is highlighted here in pink. And this encodes for the spike protein and for the RBD, the receptor binding domain. And so since I'm going to be mentioning these terms, I'm just putting them up front already now. So we have the S1 domain here, which is this part of the, right, sort of the N-terminal part of the protein, within which is the receptor binding domain. And these have generated a lot of interest naturally. The spike protein and these domains have generated a lot of interest because this is the protein that allows the virus to enter cells through binding to the ACE2 receptor. And this is also the very same target of antibodies. And right, I'll just share with you that at the beginning of the pandemic, several researchers, including me, were kind of asked, do you think there's going to be evolution, a lot of evolution that we see in coronavirus? And I thought to myself, well, there's not really a high probability that we'll see so much evolution because the same target that antibodies detect is the same, basically, right, domain that allows the virus to enter the cells. And so there's going to be purifying selection against mutations there. And of course, I turned out to be extremely wrong, as were other researchers, and there's been a lot of evolution in this region. And so again, as you know, the variants of concern till fall 2020, actually evolution was kind of pretty boring. There wasn't anything going on, and then came the variants of concern: alpha, beta, gamma, delta, and Omicron. Some of which, of course, achieved global spread. With Omicron, actually, it's like a whole suite of different variants, and there were these subsequent waves, BA1, BA2, and so on, till this very moment as we speak, where new variants come out. And sort of one thing that was noted already early on is that all these variants of concern are characterized by a lot of mutations. And the pattern of mutations is also uneven. There are a lot of mutations that are in the spike protein, a lot of non-synonymous mutations, and these are kind of signals that are indicative of positive selection. This is, of course, Omicron or BA1 when it first came out. Again, one of my predictions that turned out to be very wrong. The first time I saw this sequence, I was pretty sure this was a sequencing error. I said, there's no way there could be so many mutations. And again, I turned out to be wrong. And so this is, of course, more than expected based on the molecular clock rate of evolution. So, I mean, if you look at the evolution of SARS-CoV-2, during a wave, we see this kind of really set pace of evolution with accumulation of mutations occurring just like a clock, right? And that's what molecular clock is based on. And whenever we see these variants come out, we see this big burst of mutations that come up, just like you saw JT show with BA2.86, which is actually an extreme case in itself. And so the hypothesis, of course, emerged was that variants of concern originally emerged in chronically infected individuals where there are strong selection pressures, possibly due to the treatments they received. And so a lot of the treatments are based on monoclonal antibodies or plasma from convalescent individuals. That was in the past. Now it's more monoclonal antibodies or combinations. And so this will lead to strong selection pressure to evade these treatments. And large viral population sizes. And so during transmission, we have these transmission bottlenecks, and these are very, very stringent transmission or very, very small transmission bottlenecks. So most of the data that is coming out is showing that the transmission bottleneck size is about 1 or 2 viruses or genotypes that are transferred from one individual to another. So that's a really severe bottleneck. And we know from evolutionary biology that this will lead to a decrease in the rate of adaptive evolution of the virus. So presumably in chronic individuals where we don't have these bottlenecks or they're not as severe, then the rate of adaptive evolution might increase. So this was this hypothesis, and let's begin by defining chronic infections. So this is the definition we put forth in the first paper that we came out that we wrote, and these are prolonged infections, more than 21 days, usually months or even up to a year or beyond, where there's active viral replication. And this is kind of a key point. Okay, so there, at least in the case reports that we initially focused on, we did focus on this element of active viral replication, or some indication that there's active viral replication, as opposed to long COVID, where long COVID is a situation where symptoms persist and not necessarily also active viral replication. There is some overlap, by the way, sometimes between long COVID and chronic infection, but the way we're defining it now is just on this from the viral side. All the patients, or literally all the patients that have been reported so far, are severely immunocompromised. So there are 4 categories: hematologic cancer, leukemias and lymphomas, transplant patients, AIDS patients, and autoimmune diseases. And so both transplant patients and autoimmune diseases are treated with immunosuppressive drugs. And so this, of course, leads to an immunocompromised situation. And I'll emphasize that actually the majority of patients are from the first category, at least in the Western world. So thankfully, thanks to treatment, a lot of hematological cancer patients are able to survive for more lengthy periods. And this leads to the fact that there's actually quite a large body of these patients with a severely depressed immune system out there. Some of these patients exhibit rapid evolution and antibody escape. And so basically our first study focused on gathering case reports. And so we, at that time, thought this was a really big number that we put together. We looked at 27 case reports where there was also high quality sequencing data. And so this is a list of patients. And actually one of the things that we found was that there are these diverse outcomes. So in some cases, we see this very rapid evolution. And so this is this column here with a green background. We see a lot of mutations, a lot of spike amino acid replacements. And actually in some of the patients, we don't really see anything going on, very little evolution at all. And this was actually pretty intriguing in itself and showed that we really can have very diverse outcomes. But I'm going to focus on one just from that study, on one of the results that we found. I mean, I think that everyone kind of knows this by now, but the result was that we see these very similar patterns of mutations if we kind of aggregate all the chronic infections and aggregate all the lineage-defining mutations for variants of concern. And so the x-axis here is the genome position, the y-axis here are the counts that we see when we aggregate. And we see that in the spike gene, we see a lot of mutations. Again, we all know this is, and this makes sense from an evolutionary perspective. This is where basically what we're probably seeing most of the time is antibody evasion. And so this was interesting, but one of the more interesting findings here was what was absent. Okay, so what I'm showing you here are 2 categories of mutations, or 2 kind of bins of mutations where we didn't actually see, where we have a lot of mutations and variants of concern, but we didn't see too many mutations in the chronic infections. And then we intersected this with data that came out of Trevor's lab, where they analyzed different clades and the logistic growth of clades bearing specific mutations. And what we saw is that these regions that I marked before had specific mutations that were associated with higher logistic growth. So the very mutations that are presumably associated with higher transmissibility are the ones that are absent, or mostly absent or some of them are absent in this set of 27 chronic infections that we found. Generally, we think that we think the reason for this is that chronic infections are, the virus is less transmissible. And so there are kind of two similar reasons for this. One is that the virus, or similar or kind of partially overlapping reasons, that one of them is that the virus migrates to the lungs, and then we have less transmission. So if the virus migrates from upper airways, there's a lot of transmission. Lower airways, we have less transmission. And we also think that there's less selection pressure for the virus to transmit as long as it's so-called happy inside, circulating for these lengthy periods inside an infected host. And this is kind of corroborated by data. So to date, out of hundreds of case reports, there are only 2 really verified transmission events that are reported. There's a new outprint. There's a preprint that came out I think a couple of months ago that also documented a relatively low rate of transmission event and presumably what they find as chronic infections. And so it doesn't mean that these transmission cases aren't occurring, but it seems that they're not very, very prevalent in chronic infections. And so I'm now moving on to our newer study. So this is going from small to big. So really the caveats of the previous study was that there was a very small sample size of 27 patients. All of them were older sequences. Okay, so they were from wild type or alpha. Most were actually from wild type virus. And the big question was, how can we go bigger? And so actually one avenue would be to mine the literature again. And we tried that at some stage. And I can tell you that it's a nightmare. I know other groups in the world have been doing this. It's a nightmare because the reporting is different, and the sequencing quality is terrible. It's really hard. People call chronic infections actually different names. It's chronic, it's persistent, it's prolonged. And then actually, Shai, our collaborator, came up with what I think is a really beautiful idea, and that is to use the big data that's out there. So today there are more than 16 million sequences available on GISAID. So that's the public, the database where most researchers submit their SARS-CoV-2 data to. There are more sequences than that out there, but this is kind of the biggest database. And we thought to ourselves, some of these might be chronic infections. And so then the question was, how could we find them? How can we find them? And we focused on this idea of searching for monophyletic clades. And what you see here in this figure here is taken from a paper of Kemp and colleagues from 2021, one of the first reports of chronic infections. What we see here are 4 chronic infections. So every color is a series of samples from the same individual who's chronically infected. And what we can see here is that at least for 3 of these patients, the clades are monophyletic. So monophyletic means that the clade clusters together and there's nothing else together within that clade. There are no other sequences from someone else here. And so our idea was that we could mine this huge phylogeny for clades with the same age, same sex, same location. Okay, these are all metadata that is available in GISAID, and where the time interval is more than 21 days from the first to the last sample in this clade that we find. And so our underlying assumption is that each such clade that we find is from an individual who's chronically infected and repeatedly sampled. And I will say that one caveat already that that comes to mind with this kind of way that we're doing this is that we're assuming no onward transmission, right? So if there would be onward transmission in this blue individual, we'd see kind of a branch coming out somewhere here, and it would be a different color, right? Reflecting that this was transmitted to a different individual and the clade wouldn't be monophyletic anymore. And so this is a caveat that means that we're missing out on some data, but currently it's too tricky to take into account transmission. It's actually something we're looking into, and in future work is trying to make the model even more trickier than we did already. And so the first thing that we did, one of the first things that we did is we did a sanity check. And so we asked ourselves, what is the chance of finding such a monophyletic clade with where all the sequencers are from same age and same sex just by chance? And so this was a very hard question to answer, actually. But what we did is we focused on clades that were sampled up till where all the sequences were up till March 31, 2020. So very, very early stages of the pandemic. And we assumed that there was probably not enough time for chronic infections to be detected at such an early stage, right? So if we're looking at the timeline of the pandemic, this graph here on the bottom, right, this is a very, very early stage of pandemic, and we found 0 clades that have same sex, same age. I actually forgot to mention that one of the requirements is that we're requiring at least the clade size has to be at least 3 sequences and above for us to take it into account. Right, so this was partially reassuring. And I'm going to go into the methods and numbers that of the data that we use. Okay, so we began again with a GISA database and Usher tree. So Usher is right. They constantly maintain this huge tree of all the sequences, all the SARS-CoV-2 sequences. So basically, we began with 11.7 million sequences after extensive quality control. And unfortunately, only 2.9 million, so that's 25% of the data, were sequences with valid metadata, so age and sex. And so already at this stage, we were kind of disappointed. Why only a quarter of the data? Why do we have to throw away 75% of the data? But remember that 75% because we're going to get back to that later. And still, the good news was that 2.9 million sequences is still a huge number to start off with. And if already this is kind of a call, because I think that a lot of the metadata was missing because people who submit to just say sometimes aren't aware that it's important to add this additional information on. And I think it actually is for a variety of different scientific reasons. And second of all, perhaps there are issues with fear of identification. But I really think that there's no way to identify a person just based on age and sex, given this huge volume of sequencing that is out there. We looked at a set of negative controls. So basically what we did is we sampled clades that are not the same age or sex, but are from the same location and kind of span a similar size of sequences and similar timeframes. We kind of try to match the clades that we found I'm going to show soon that we found so that we can have a proper control. We found 18,000 control clades, and everything that I show you is going to be a bootstrap over these control clades. And we had our set of positive control, which are real chronic infections. And these are 32 published case studies with high quality sequencing data that we could use. And so we found 271 of these monophyletic chronic-like clades, right? I'm going to be calling them chronic-like from now on. And so this is their distribution across the different variants. And so it was reassuring to see that we're seeing, a good distribution from the different variants. And naturally we have, for example, more BA1 because there are more BA1 sequences. And the big question was, are they really from chronically infected individuals? And so I'll already say that, we're never going to know if we're not going to be able to track every one of these down and say, is it for sure chronically infected individuals? But I hope I'll convince you soon that we think that they're enriched, that the set of clades is enriched for chronic infections. And so, first of all, we looked at the distributions of age and sex. And so what you see here is that the controls, which are in dark blue, have lower age. There's a lower age distribution as compared to both the chronic-like clades that we're finding, these 271 chronic-like clades, and these are the real chronic infections. And also, we're seeing a higher proportion of males in the chronic-like and real chronic infections. And we think the reason for this actually really makes sense. So the first reason is when we look at the categories that are of immunosuppressed individuals, hematologic cancers, as I mentioned before, is the largest category. So older males tend to be sick with hematologic cancers more than females and more than younger individuals. And so already that made sense. And the second reason is, again, severe COVID itself, right? So hospitalization rates increase if you're older and more male. And so it, to us, made sense that, right, if we have a chronically infected individual, and he or she are hospitalized, then there's also a higher probability that they'll be sampled repeatedly, and we're going to see this in the database. And so this kind of made sense. The second indication that we looked at was at something called the Sacken index. And so the Sacken index is a measure of tree topology, of the shape of a tree. And so there are two subtrees that I'm showing you here. The top one, right, we see this kind of fork shape, right, which is also called a star-like tree topology. And the star-like tree topology is actually something that we see a lot in the SARS-CoV tree, all along the tree. It's usually thought of to be a sign of a lot of super spreading, right? So a super spreading event where one person suddenly infects a lot of people will look like this, will have this star shape. And this is associated with a lower Sacken index. Okay, so community spreading, lower Sacken index, whereas when we see intrahost evolution, when a virus evolves within a host, we usually see this ladder-like topology. Researchers from the field of HIV are very familiar with this. We see this kind of ladder-like topology, which indicates that we're seeing kind of lineages replacing one another sequentially in time. So this is associated with less higher Sacken index, and we see this the statistically significant positive signal where our chronic-like clades have a higher Sacken index. So this kind of shape, intrahost evolution. And the next line of evidence that we looked at is we looked again at the same kind of figure that I showed you before, where the x-axis is the genome and the y-axis are the counts. We're trying to look at the pattern of mutation along the genomes. And again, for the chronic-like clades, similar to the real chronic infections, we're seeing a lot of enrichment in the spike region, and especially at S1 and especially at the receptor binding domain. And so there's similarity between these two. In the control clades, we see much less of this. I will note that there's one bin here in the N-terminal domain where we do see enrichment of mutations in the control clades. This is something that actually appeared later in the pandemic. At the earlier stages of the pandemic, we didn't see this enrichment at all, but kind of somewhere in between BA1 and BA2, we started seeing this N-terminal domain enrichment also in the control, but nothing in the other domains of S1. And I think this is kind of interesting. We might be able to discuss this later. But the main message that I think is really clear from here is that we're seeing similarity between the chronic-like clades and the real infections, and we're seeing dissimilarity between the chronic-like clades and the control clades. And this leads me to conclude that these clades are likely enriched for chronic infections. And so I'm going to just show you some example. This is a clade. All the green dots here represent sequences from an individual from Spain, Madrid, male, age 57, sampled between March 7, 2022 and April 28, 2022. And when looking at this clade, sort of people that are familiar with mutations might be able to really quickly pick up, right, that there are familiar mutations going on here. So these two mutations highlighted in blue are in the spike. These are monoclonal antibody. There's a therapeutic called sotrovimab, which was very, very popular and used during the BA1 wave. And what we're seeing here these are very well-known escape mutations from this therapeutic. And then there are these two mutations highlighted in purple, and these are mutations that we actually saw previously in that set of 27 chronic infections that I showed you before. We saw these mutations coming up a lot. And I'm going to get back to these mutations a little bit later, so keep them in mind. This is another example. This is one of the most extreme examples and one of the most divergent examples that we had in our data set. So you see that there are like 37 amino acid mutations here, and there are a lot of spike mutations going on here. And so this was, again, All of the samples here are Slovenia, female, age 61, and the dates spanned this quite a long time range. Right, the next thing that we did is we wanted to somehow be able to look at the rate of evolution and to ask if we can find actually a sort of more robust way to measure adaptive evolution going on here. And so what we did here was we performed root to progression of each one of the clades, right? The number of mutations along time in each one of the clades, and we compared the chronic-like clades to the control clades. Now I'm gonna already say that there are a lot of caveats to this analysis that we did. I'm aware this is a problematic analysis. The major problem is that there's a lack of temporal signal, right? So these are very short timeframes that we're looking at. And so in a lot of the clades, we don't really see right, a significant kind of slope. Other problems that there are is that there's incomplete purifying selection going on. So when we look at the end of the tree, we have a lot of mutations that are kind of are there, but will probably be weeded out by purifying selection. So this is adding on noise to the data. In chronic infections, we know that there are subpopulations of data, subpopulations of virus. So this is something that we actually have seen both in our previous study and in this study, where it seems that that there are mixed populations of virus co-inhabiting a patient at the same time. And so this is again causing problem when we're trying to perform this root to progression. But our hope was when we originally performed this, that if we average out enough data, then we'll still be able to get a signal. And so I'm sharing with you our results despite all these caveats. And so these are the average slopes when the control is compared to the chronic-like infections. And so whenever we look at non-synonymous mutations, we always see a significantly higher number in the chronic-like clades as compared to the control clades. We don't see this higher rate in the synonymous mutations. This was a good sign because we don't expect the rate of neutral evolution to change between our chronic infections and our control clades, which represent transmission in the general population. And the highest differences are observed at S1. And so we see a 2.5 to 4.5 higher slope and spike S1 in the chronic-like clades. And then we went on to focus actually on a very specific set of key RBD mutations. So these are the mutations that are seen very, very often in a lot of variants, and the BA1 variants and the BA2 variants. And we asked, what is the probability of observing these BA1, BA2 mutations? And you can see that it's times 20 higher in BA1 chronic-like clades, it's times 10 higher in BA2 chronic-like clades. So it really does seem that what we're seeing is in these chronic-like clades, the rate of adaptive evolution, or at least this proxy that we're using for rate of adaptive evolution is really higher. And so the big problem was that this was actually only 25% of the data, right, as I mentioned before. And so our big question was, is there any way we can use the additional 75% of the data, which is really a really big number of sequences, and somehow extract more information from these sequences? And so to this end, what we did is we used large language models, right? These very popular models. And I'll show you why Danielle Miller, the wonderful author of the study, actually, she convinced me that this has to be the way to go about when looking at such big data and such noisy data. And so our idea was that we can look at a sequence, and we look at the sequence, and it's a SARS-CoV-2 sequence, and we always compare it to a reference sequence, which is the Wuhan sequence, the first sequence of SARS-CoV-2 that was published. And so this is just our useful comparison as a starting point. And what we can do is we can define words as mutations as compared to that reference genome. Okay, and then the sequence can be collapsed to a string of words. Okay, and the string of words is basically like a sentence. And this is what language models do, right? They can analyze sentences. And our idea was that some of these words can allow us to predict chronic infections. So maybe there are words that are very unique to chronic infections, and these words are absent in or less prevalent in the general population. or they're enriched for in the chronic infections, and they're less prevalent in the general population. And so this was the kind of the assumption that we set out to. And I think the second important assumption to mention here is that context matters. And this is something that is important in language models. The context matters. And so the context matters. And this is right. This is true for language, right? You use a specific word, and it's kind of important what the words next to it are in order to determine the meaning of that word. And so for corona, the context matters because it matters if we're looking at a background of a Delta variant versus a BA1 variant versus an Alpha variant, right? That's the context, and it matters because different mutations have different meanings in this context. And so we trained a classifier basically that can predict chronic-like versus control. And so some of the more technical details are here on the right. And so these are our initial results, right? We found words that are associated with chronic-like clades. And so these are these words, and right, so these are long lists of words. There's a lot of mutations that came up here. And again, I'll just emphasize these are not mutations that are absent in the rest of the data, right? And the sort of the sequences that are represent transmission. But they are more prevalent in chronic-like clades. And so some of you that are more familiar with the mutations, you'll notice that there are a lot of mutations that pop up here that are, that we're very familiar with from one variant, from another variant. And I'll already say that most of these chronic words are actually associated with high fitness, right? So this is data that we took from a paper from Jesse Bloom and Richard Neher, which inferred the fitness of every single mutation in the context of the general population, so from transmission chains. And so most of the mutations in their data were inferred to have very low fitness. And this is something we know from evolutionary biology. But the mutations that we found, most of them are actually high fitness. And so this was reassuring because it means that these are kind of mutations. It makes sense, right? That we're going to see mutations in these chronic infections that are giving the virus higher fitness. And again, most of them, and so the top panel is all the genes, the bottom panel is spike genes. And in the spike genes, we see mutations, mostly mutations associated with antibody evasion. And so presumably these are mutations that allow the virus to evade either therapeutics or the kind of weak partially functioning immune system in these chronically infected patients. And these are mutations that are also associated with higher fitness in the general population because, right, we have these subsequent waves and immune evasion. The interesting kind of point here was that we actually saw two mutations, or we saw several mutations, but I'm highlighting two. We saw several mutations where the opposite occurred. So these are mutations that have very low fitness in the general population, but are very, very prevalent in chronic infections. And so I'm highlighting two of them, ORF1A, T1638I. And so we actually saw it right in that Spanish patient that I showed you before. Another one is envelope T30I. And so both of them are actually prevalent against different backgrounds. So this first mutation here is prevalent on alpha, delta, BA1, all of the backgrounds that we found. And so it kind of seems that these are mutations that are kind of specialized to operate, if we can say operate, in the context of a chronic infection. And so we kind of tried raising hypotheses specifically as to these two patients. So the first mutation, this ORF1A mutation, is a mutation that was actually shown to be associated with cytotoxic T lymphocyte escape in the context of a certain HLA background. And so we thought that maybe in the general population where we have a whole lot of different HLAs, maybe there's a fitness cost for this mutations against a different HLA background. And so once this virus is transmitted, there's going to be a fitness cost and the mutation will disappear. And the envelope mutation is actually a mutation that has been noted in serial passaging in particular cell types and not in other cell types. And so once again, our hypothesis here is that maybe this mutation allows for an advantage in lower airways as compared to upper airways, or perhaps other tissues. And again, these are just hypotheses, but I think this is a really interesting avenue to follow. I mean, looking at these mutations that are really particular to chronic infections. Then the next stage we performed is actually we use these words to predict more chronic-like clades. And so the graph I'm showing you here, with like every classifier, depends on the threshold, right? I mean, the number of clades that we could predict depend on the threshold. And we actually believe that the threshold probably should be around 65%. I won't go into the details, but this gives us almost another 1,000 clades to look at. And this is something that we're looking at now, trying to find kind of more insights from these additional 1,000 clades that we find. I'll just kind of highlight two of these clades. The one was kind of exciting. We were looking at this clade. We were thinking, is there any way to validate this? I mean, do we know anything about this? We looked at the submitters of the data, and then we actually noted that we'd been in contact with these with the people who had submitted this. We contacted them, and they validated that this is a chronic infection from the Netherlands, but they had uploaded the data without sex and age. And so this is a real chronic infection. And this is just to highlight, right, that of course, most of the data that we have out there, there's unknown sex, unknown age, and we have no idea if this is a real chronic infection or not. But I am excited because I'm pretty confident that a proportion of these are real chronic infections and that we can learn a lot more from looking at them. The last bit of data I'm going to show you is how we use these words, these mutations, to predict the future in hindsight. Okay, so this is a timeline of the study data, and so we downloaded the data during September 2022, more than a year ago, and so basically all our clades are from sequences that were sampled before September 2022, and we asked ourselves, can we use these mutations to predict the variants that are circulating at present? Okay, so September 2022 till today. And so we focused on 14, so the 14 RBD mutations that come out of our chronic-like clades. These are the ones that are labeled. There are only 14 spike RBD mutations that are labeled by our language model as so strongly associated with chronic-like infections. And then we kind of looked at different measures of of their success in the future. I'll just highlight one of these measures. So maximum global prevalence. We looked at their maximal global prevalence after September 2022. Okay, so in this gray shaded area. And you can see that some of these mutations achieved really high frequency, 100%, 94%, and so on. So 9 of the 14 reached a global prevalence of higher than 1%. And I'll actually mention that even some of the ones that are mentioned here as lower, right, for instance, 6.9% or 2.8%, if you stratify them by different regions in the globe, so in some regions of the globe, they reached higher prevalence. And so I really think, this to me shows that these mutations that we're finding in these chronic-like clades do have predictive power. Of course, this is not, right, the only measure we can use for predictive power, but I'm really excited because I think that the way influenza vaccines are updated is that different methods are used to predict what's going to be the next variant, and that is used to determine the next vaccine strain. And so I'm excited because I think that this is perhaps one measure that we can also use to sort of predict things that might come up in the future for SARS-CoV-2. And with that, I'll end, and I'll just summarize and say that, chronic infections are very rare. And I'll even emphasize chronic infections are rare. And I think that chronic infections that lead to onward transmission are even rarer. And chronic infections that lead to global transmission are exceptionally rare. But, if you multiply a very, very low probability by a very large number, which is a number of infections out there, so eventually you do see very rare events happening, right? Like we're seeing now for presumably for BA.2.86. So what we saw here is that they might speed up evolution. And I think another really intriguing kind of follow-up is that they might actually also allow crossing fitness valleys, right? So we might have these epistatic interactions that occur during these prolonged infections that might allow crossing the fitness valleys. This is something that we're looking at, the 2 researchers that I kind of mentioned up front that are unfortunately very traumatized now, that is part of their research project. And chronic infections coupled with language models can perhaps teach us to some extent to predict the future. And with that, I'll end and really thank you for your attention and thank the funding agency and our collaborators at Tel Aviv-Sourasky Medical Center, and I'll be happy to take questions.
Thank you so much, Addie, and thank you for presenting despite the circumstances. So if anyone wants to just raise their hand for a question, whether it's in chat or otherwise, please do so, and I'll call on you. Otherwise, I'll kind of work through the chat and moderate these questions. But please raise your hand if you'd like to ask something verbally. Kind of working through chat. First, let's start with Josh. Your hand is raised.
Oh, Trevor, you can go through the chat first if you'd like.
No, let's start here.
Great. Thanks, Adi. That's just a wonderful talk and and so thought-provoking. I'm still stuck on the transmission bottleneck because when we see these people in clinic, they're often not terribly symptomatic, but their viral loads are extraordinarily high in the upper airway. And coupled with the fact that there's also been this work in wastewater showing viruses that look strikingly like Omicron that never sort of left that likely single person who is shedding the virus. And I'm just wondering if you could elaborate on that a little bit and where you think the largest bottleneck is that prevents more of these viruses from spilling into the population and that these events really are, despite their massive effect, quite rare. Thank you.
Yeah, I'll just say that I think it's an excellent question. I don't think I have a really good answer on that. It's something that we've been struggling to understand as well. I'll say that the patients that we had been looking at, what we saw is these cycling patterns of viral load, right? So sometimes the viral load is very, very high, but then it drops and the virus disappears sometimes for weeks and even months, and then it comes back. And so I think that in this intermediate stage, it's migrating to lungs or somewhere else. And sometimes when there are samples, right, from like these whatever way there is of sampling from lower airways, and the medical staff that we've been working with did see positive results. But yeah, I am struggling myself to understand if it's really only because they migrate to the lungs, or as I kind of mentioned, because they're lacking specific mutations that allow them to transmit. I'm really not sure. So yeah, I think there could be a lot of different reasons. And it's a great question. I think we really do need to focus more attention on understanding it.
Great, thank you. Okay, so I'm going to work through the chat a little bit. So Cornelius asks a couple of questions about metadata. The first is maybe just looking at geography, like detailed geography, and like sequencing lab would be enough to think that this is a chronic infection. Wanting to know if you're accounting for plus or minus 1 year in terms of age differences, because people age over time. And then kind of in related vein, how much could you imagine contacting sequencing labs to check to see if they kind of know these are chronic infections actually, and whether there's additional metadata in that case for these things that are being flagged?
Yeah, so they're all great questions. Can you remind me of the first question, because I had that in mind?
Yeah, the first question was detailed geography and
Right.
whether you really need age and sex, and whether you could rely on something coarser.
Yeah, so we do look at detailed geography, right? We look at the lowest level of resolution of the detailed geography that we have, and we do enforce that. We enforce the same exact location. Regarding sequencing lab, I think I don't think we could use only that, because, for instance, if we have a sequencing from an outbreak at a hospital or a school or something like that, it might be the same sequencing lab. And that was actually our biggest concern. So the biggest concern that we had, and the reviewers had as well, and I think justifiably, is that what we're picking up might be an outbreak in a specific kind of setup. I think that looking at this a lot, I don't think that it's very likely that we're picking up too many of these outbreaks, because requiring the same sex and age, exactly the same age, would rule that out. With regards to age, yes, we do allow for plus minus 1 in age, and then we go back and look at it sort of more carefully. So yes, we do take that into account. And again, I forgot the third question. I'm sorry.
The third question was about recontact, and then yeah, following up with
Oh yeah,
the
recontact.
labs. Yeah.
Yeah, so like I mentioned, there was this one lab in the Netherlands that we had contacted. But again, these are big numbers that we're looking at, and it's really hard to contact. We actually had thought of taking all the Israeli cases and sort of because these are a lot of times people that we're in contact with as it is. We do have an issue here with privacy, so we're kind of concerned that we don't want to sort of right, step on anyone on that way, and so we're kind of avoiding that avenue at the moment.
Great, that makes sense. Thank you. And so moving down, question from Christian about whether these methods can be used to look at other viruses, influenza, dengue, et cetera. Also kind of, I'll piggyback a little bit there of, it's my impression you can find this with flu, but not nearly to the same degree. And kind of, do you have guesses or insights into maybe biological differences that might be making this more of a unique thing for SARS-CoV-2?
Yeah, so these are great questions. Again, questions that I think I and many others have thought of. I will say I had a kind of very quick attempt to try and look at flu. The biggest problem there was metadata or lack of metadata. And so I think once there isn't this metadata, and right, the numbers already dropped dramatically. We're not in the tens of millions, we're way less, and then the lack of metadata so it makes it very tricky. But I do think it's possible of thinking kind of alternative ways, right? So I mean, originally, actually, when we started off the study, we tried looking at sort of long branches just as indicators. We actually found out that that's kind of too much of a noisy method, but so we looked at the monophyletic clades. But I do think that it is worth looking at other viruses and trying to think of methods. And I think it's kind of an enigma why we're seeing it so often for SARS-CoV-2. I mean, for me, the biggest answer was just the numbers. Okay, so once it's a very rare event, and you have to get very large numbers of infections to see it happening. I was, however, surprised when BA 2.86 came out, because in my mind, the numbers had dropped. So I didn't see it didn't completely make sense, but it is possible, right, that it's because it's from a much earlier stage where the numbers were higher. So I'm not completely sure. Yeah, so that's kind of my attempt to answer that.
No, that's great. Almost slightly on the same lines, maybe kind of. So I've been very curious as well about the wastewater lineages. So you can kind of identify these cryptic lineages. They have all of these mutations. They look like they look a lot like chronic infections, at least kind of anecdotally. Have you, with your kind of more careful work here, have you done a head-to-head comparison of the mutations that you see in wastewater cryptic lineages to these chronic, like bona fide chronic infections, and then also these chronic like clades, to see if they seem to be the same thing or they seem to be different?
Yeah, that's something that I did look at some stage, and my answer is that there is some overlap, but not a lot of overlap. I actually expected a lot more overlap. So there are some like key mutations that have been flagged by wastewater sequencing labs, and we didn't see them here. And so the top mutations that are flagged in the wastewater are not observed in our data set, but the sort of lower mutations are observed in our data set. Again, not sure what to make of it. I think again, the problem might be with our sample size. So again, it keeps going down to the sample size. I mean, we started with 27 and it was too little. We went up to 271. It's still not big enough. And so actually some of what we're working on now is to try and reduce. I mean, so we were very stringent the way we worked here, because we were so scared of picking up false signals that we were very stringent. And I think that the language models and reducing the stringency might really allow us to get a much larger data set, and then we might get a better intersection with wastewater.
Great, thank you. Cornelius, go ahead.
Oh, and I'm sorry, I'll just add one last remark, is that I think maybe one of the differences with wastewater is that one of the hypotheses is that wastewater is associated with mutations that allow replication in the gut or something to that effect. And whereas what we're seeing is most often samples from upper airways. And so again, that might be a difference.
Okay, yeah, that's super interesting. And then like, sorry to that like you could imagine maybe these chronic infections upper airways are the things that are actually spilling over occasionally with BA.2.86 and so forth, and the more gut associated wastewater lineages are not. Yeah, seems super interesting.
Yeah.
Cornelius, it looks
So-
like you
so-
briefly
so my-
had your hand. Yeah, go ahead.
Yeah. My question was about sort of the types of immunosuppressed people that might have chronic infections, because in the West or in rich countries, it seems to be mostly people who take immunosuppressants. And the VOX that we think come from chronic infections, BA1, BA2, potentially BA45, BA275, BA286, most of them actually seem to have first spilled over in Southern Africa or India for the case of BA275, which might not be where people take lots of immunosuppressants, but where instead they might have chronic infections because of HIV infections or other things. Now, I was wondering, did you manage to sort of get any chronic infections from those sorts of countries? Because we know that there are lots of chronic infections in rich countries, but they never, apart from maybe B117, seem to cause any transmission. It might be a different that HIV is a better breeding ground for HIV chronic infectees are a better breeding ground than
I think that's an excellent question. I think that's a really, really good question. My short answer is that we are definitely enriched for Europe and US in these 271 clades. We saw that very, very clearly in the data. And so no, we don't have a lot of data. In the 27 original sort of set that we started off for, we did have 2 AIDS patients, 2. But that's 2. And I very much agree with you. I think it might cause the immunosuppression there is really different, right? It's coming from cytotoxic T cell depletion, right? So it's really different. And I agree that it could cause something different, but we don't have enough data to look at it.
Yeah, thank you. Okay, so this is going to be the last question. We're slightly over time. And then I'll combine this from Lauren and Josh. If the immune pressures are so different in an immunosuppressed patient leading to these distinct mutations, is it surprising that they seem to have predictive value for the mutations generated in the general population? And they seemed related enough from Josh. Is there any sense whether there is a unique niche of immunosuppression that provides the greatest risk for generation of VOC-like viruses? Even, which I'm thinking of as viruses that are going to spread in the general population. Even within severely immunocompromised population, there's a gradient with almost no B/T cell pressure to all versus immune selection pressure that may be insufficient to clear the infection or strong enough for selection. Does that seem fair? Go ahead.
Right. So yes, I agree that there are different selection pressures in the patients, but I was actually surprised by the fact that we did see that most of the mutations are high fitness mutations in the general population. So it means that there's some difference, but it's not a huge difference. Okay, I think that's what our data is showing, and that's why we are able to show this predictive value. Our predictive value is only for RBD. Okay, that's what we focused on. That's I think we kind of naturally focused on that because a lot of variants are kind of defined by the RBD mutations, but there's much less predictive value if we focus on other regions in the genome. And I think that's kind of the interesting take from this, right? I mean, there's been so much focus on RBD and on spike and so on, but there are other regions of the genome that are teaching us sort of different things about how the virus is evolving. And I think we are very interested in these other regions, but the problem, it always boils down to the fact that we don't necessarily know what to say about these other regions, except sort of hand wave. And so, yeah, that was regarding that. Again, managed to forget the second question. This is happening to me all the time today. I'm sorry.
I think similar question. So, any sense there's a unique niche for immunosuppression that provides the greatest risk for generation of VOC like viruses?
Yeah. The shorter answer is that I'm not sure. Again, first, I think it's a one in a. The way I see it is actually it's probably a probabilistic event, just a 1 in a million or something like that event that happens in an immunosuppressed patient. Again, probably after a long time where we also have this ability to cross fitness values right through epistatic interactions. I think this is what is generating. I mean, again, but this is kind of more of an educated guess based on what we saw in Omicron and now in BA 2.86, these very, very long branches. In our data, we didn't see any, or we couldn't say anything specific about niches.
Okay, no, that's wonderful. Okay, we'll close there. Yeah, thank you so much, Adi. That was an excellent talk. Helen, do you want to mention, yeah, next month?
So next month we have Professor Iwasaki from Yale and HHMI who will be speaking to us about long COVID. So we look forward to seeing everyone and Happy New Year.